So I have been getting a lot of questions about how independent and dependent assortment work, especially how dependent works when the genes are linked on the same chromosome. So to understand this, we're going to look at independent and dependent assortment in meiosis and figure out the difference between the two. So let's imagine we have a female that's all homozygous dominant mating with the male that is all homozygous recessive. The F1 generation will always be heterozygous. Now, how do we draw this on chromosomes? Since there's two different letters, we would need two different size chromosomes. We would have, this one and this one. And it also needs a homolog here. And this one is from the dad. And this one's the mom. So during meiosis, these homologs line up next to each other, then they both separate and move towards the two poles. And it's also important to remember that sister chromatids form that are identical. So after these two homologous pairs separate towards the two poles, they divide again and gamete formation occurs. So the first gamete is going to be this lowercase and this lowercase. Then we have this lowercase, this lowercase. Now on the other side we have capital, capital, and capital, capital. So even though 4 different gametes are produced, these two would make one combination. These two would be the second combination. This is dependent assortment. This is because we assume that when these two chromosomes line up, all the capitals end up on this side of the line, and the two lowercase are on this side of the line. And in the parents, all the capitals were together and all the lowercase together. So these two assort dependently of each other. All the capitals are always found on the same gametes. So this only produces two combinations, all capital and all lowercase. Now, let's look at how this is different from independent assortment. So going back, dependent assortment would produce only these two gametes. Now, although this is a very likely possibility, you can also have another equally possible formation or assortment, and that would include the capital A, imagine the two B's switch. So the A's can stay the same, but in the B's, the lowercase b ends up on this side, the capital B ends up here. Now both of these are equally probable. Now if this were to happen, these two would go to separate poles, then they would divide again. And look at the gamete formation here. These two would produce lowercase capital. These two would produce lowercase capital. This one would produce capital lowercase. These two would produce capital lowercase. Now, independent assortment states that the gametes can either line up this way or they can line up this way. So in independent assortment, these are allowed to switch sides so that the lowercase and capital don't have to stay together. So the different gametes that are possible are capital, capital, lowercase, lowercase. You can also have capital, lowercase, and lowercase, capital. So while in dependent assortment, you only had these two choices because the capitals had to stay together and the lowercase had to stay together. In independent assortment, you get 4 different choices of gametes. Now, I'm sure you've always heard of the ratios 1 to 1 to 1 to 1 and 1 to 1 to 0 to 0, but where do these actually come from? So let's go back to our original parental and F1 generation, and let's say we're doing a test cross. And we're doing the F1 female, which is this, and let's mate it with the original parent. Okay, now let's list all the possible gametes for both of them. This, the only gamete it can produce is lowercase, lowercase. However, this one, if we go by independent assortment, can produce 4 different gametes. Capital, capital, lowercase, lowercase, capital, lowercase, lowercase, capital. If we were to do a Punnett square, using independent assortment, we would have AB mating with a AB, 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 and AB. This would give us this choice, this choice, this choice, and this choice. In here, the F2, all 4 of these have 4 different genotypes. So the ratio would be 1 to 1 to 1 to 1. Independent. Now let's assume that dependent occurs. What did we say were the possible combinations of gametes if dependent assortment happened? Again, this would only have AB, that stays constant. But remember, in dependent assortment, the parents would have to stay together, so there'd only be 2 choices here. So then we have an AB mating with an AB, an AB. These 2 would not be possible using dependent assortment. So it would, these 2 are just not possible. So you could have an AA, BB, AA, BB. These 2 aren't possible. So the ratio here for the F2 would be 1 to 1 to 0 to 0. This is the ratio for dependent. Now to learn about recombinant genes, we're going to use Morgan's second experiment. We start with a wild type Drosophila fly mating with a double mutant. And this one was the female, this one was the male. Now the way that wild types and mutants are labeled is you take the letter for the mutant and you keep them the same. But then for wild type, you use those same letters, but then add a plus. And this is the parental generation, and we're saying they're purebred, which means they look like this. Now, we all know in the F1 generation, all the flies look the same. They all looked like the wild type. Now, Morgan did a test cross where he took a female from the first generation and mated it with a double mutant male. So this would be a heterozygous mating with a homozygous mutant. Again, to make this Punnett square, let's list all the gamete possibilities. We only have this one for the male, and then for the female, we can have B plus, V plus, BV, BV plus, VB plus. So our Punnett square would look like a BV, and you would have a B plus, V plus, BV, BV, B plus V, V plus B. So you can have a B plus B, V plus V, B, B, V, V, B plus B, V, V, B, B, V plus V. So independent assortment, which Mendel believed in, would give us a ratio of 1 to 1 to 1 to 1. However, Morgan's actual experiment gave us data that looked like this. The two wild types had 965. The completely mutant had 944. The gray and vestigial had 206. And the normal but black had 185. Now, this ratio doesn't follow 1 to 1 to 1. So it did not follow independent assortment, which, according to Mendelian genetics, it should have followed. But what could explain this? So how about we try using dependent assortment and see what our ratio would be. Using dependent assortment, we would have only two choices here, B plus V plus and BV. And we would have only one choice again for here. And the Punnett square would look like BV mating with a B plus V plus and a BV. That would be our first one. That would be our second one. So according to dependent, the ratio would be 1 to 1 to 0 to 0. Now, if we look at the actual data, although it doesn't match dependent exactly because some of these are still produced, it more closely matches dependent than independent. So, we're going to say that dependent assortment occurs. But we saw that through meiosis that the genes should be able to line up in any possible order. So, dependent would mean that they are linked. Also, that they're found on the same chromosome. So that now that we know that these two genes genes are linked on the same chromosome, and the two mutants are found on the same chromosome, we can draw those two chromosomes. This would be the B plus. This can be the V plus, B, V. These are the two gametes that are linked for the F1. Now the male only has a B, V, and the other chromosome would look identical. B, V. So apart from these two gametes that are produced, we somehow had to find a way that B plus ends up with this V, and this V plus ends up with this B. Because although this is more common here, these two are still being produced. So somehow we have to produce these gametes. So can you think of some way that these two alleles end up switching? During meiosis, they can switch during crossing over. So we said that B plus and V plus are linked, so they're very common. As we saw in the number was a lot larger than the other two, and normal BV are very common. So these we're going to call the parental. However, we said two other gametes are also possible, which only happen during crossing over. That's B plus V and B V plus. For this to be produced, crossing over has to occur in this white space and cut these two genes. But what is the actual likelihood of that being cut right in the middle? It's super rare, right? And when these are cut, these new gametes that are formed, they don't look like the original parentals, right? So these are called recombinant. And these recombinant types are super rare, and these are very likely to end up together. And if we go back to our numbers, again, the parental types are very common, and the recombinant types are very rare. So it almost matches the 1-to-1 to 0-to-0, and we can say pretty much matches, which is why they're dependent or linked. And the reason that we have some numbers here instead of zeros, even though they're very tiny, the reason for this is because they're the recombinant ones, super rare, and happen during crossing over. Now we know that linked genes are close together on a chromosome, but how close are they? The closer they are, if we look here, the white space, the closer these two are together, the smaller the white space. The smaller the white space, the less space there is for it to cut during crossing over. So the closer they are, the more rare that the recombinants occur. The further apart they are, if they're on like opposite sides of the chromosome, the larger this white space, and there's more places for it to be cut during crossing over. So more likely to get the recombinants. Now to find the exact distance between two genes, we can use percentage or the frequency of recombination. And this is calculated by writing the number of recombinants in your data over the total number of progeny, or like offspring. So let's go back to our Morgan 1 experiment. Here we said that this were the parentals, and this one were the recombinants. So we have to add (206) plus the 185, put it over the total. I'm a human calculator, so the total of this is 2350. Now the percentage, or the decimal we get, is 0.1663. Now in percentages, it's 16.6%. Now Morgan actually named this distance after himself, so we can take the 16.6 and write it into centimorgans. Now according to Morgan, one chromosome is 1 Morgan. So this would be equal to 16.6 centimorgans. Now it's important to remember that a Morgan is an arbitrary value. If I have a smaller chromosome here, this would still be 1 Morgan. If I have a larger chromosome here, this again would still be 1 Morgan. And this is how you find the distance between two genes so that you can draw a gene map. B plus, V plus, and the distance between these two is 16.6 centimorgans. Thanks for watching. I hope these videos help you and understand bio a little bit better. The concepts that we went over were independent, dependent assortment, how to find the ratios, crossing over, and recombinant genes. Have a great holiday season.